One of the really big challenges facing vascular surgeons is wound care. It's an unrecognised problem, or a problem that we don't deal with very well. We're excellent at undertaking complex peripheral interventions. We have a huge number of tools at our disposal, be it angioplasty balloons, atherectomy devices, stents, and enormous amounts of resource are spent debating whether we should use balloon X or atherectomy device Y. What we don't do very well currently is that we don't then assess how well we've done that procedure. You can have the best atherectomy device in the world. If you do the procedure badly and you don't re-establish good blood flow into the microcirculation of the foot, you're not going to improve that patient and you're not going to heal that wound. What we've been looking at is a portable tissue monitoring system that gives you real-time feedback in the cath lab about whether you have really improved the blood flow into the microcirculation of the foot. And it's done that without interfering with workflow in the cath lab, and it's also been very easy for the patients to tolerate. There's been no problems with it from that point of view. The technology works through a laser-based system. As the blood flows through the skin, the moving blood cells scatter the laser light. And depending on how much laser light is scattered, that is reflected in the signal that you can see on the monitor that you have in front of you in the cath lab. One of the great strengths of this system is that within 30 seconds of blowing up an angioplasty balloon, you can see a dramatic fall in perfusion. And then within 30 to 60 seconds of opening up an occluded vessel, you see an immediate increase in flow. And we can give that measurement a ratio, and we can look at how well the perfusion was to start with and how good it is at the end. And that increase allows us to try and predict wound healing. So we have two parts of our study, and we've been looking at validating the system in a group of relatively healthy patients. One of the things that we know is that when we measure perfusion in the outpatient clinic, we often use ABPIs or TBIs, but these are very variable between different clinicians. And one of the data sets we've collected here with this novel system is its reproducibility, and it's highly reproducible. The system simply involves applying a small adhesive sensor onto the foot and then turning the machine on. And so there's almost no variability between our different clinicians who used it. The second data set is really the first in-man clinical data. And this is primarily a safety endpoint driven study. We're interested in, are there any effects from applying the sensor to the skin? But we're also looking for clinical signals that are going to allow us to power those bigger clinical studies that we know we're going to need to do over the next year or two to really get this out into the market.